Okay, Bio30s, here is our last video on meiosis. And this one is just going to summarize everything we've done. We're going to compare mitosis and meiosis, look at similarities and differences, and then just kind of review those important things that lead to diversity. So mitosis, as you can see, is just one set of divisions, whereas meiosis, there's two, meiosis 1, meiosis 2. And at the end of mitosis, you get two daughter cells that are identical to the parents. They have the same ploidy. So if you start with 2N, they end up with 2N. Whereas meiosis, you end up with half the amount of chromosomes. That is a reduction division, of course, that happens in meiosis 1. And so moving on to this next slide, it talks about the fact that the point of mitosis is for making cells to grow, repair. So body cells, somatic cells. So if you have a shower and you shower off and a bunch of skin cells just came off on your towel, well, now you have to replace them. Mitosis, think of it's happening in my toes to make those nails grow faster. On meiosis, on the other hand, we're producing gametes for sexual reproduction. And there's important events that occur in meiosis to increase diversity, so crossing over and independent assortment, and neither of those are happening in mitosis because you're making identical offspring. So I've queued up a video for you here, and again, this is a McGraw-Hill one, and it is showing the similarities and differences between meiosis and mitosis. So let's follow along and listen.
DNA replication occurs. In both meiosis and mitosis, the nuclear membrane breaks down as the DNA organizes into chromosomes. In meiosis, chromosome pairs come together, or synapse, and crossing over occurs, resulting in mixing of the genetic information between the chromosome pairs. The paired chromosomes that...
So, of course, we know that that is happening in prophase 1, and that's our first really important event that is going to contribute to diversity. It's not happening over here in mitosis, right? Mitosis, we're making identical cells, so we're not talking tetrads. We're just looking at regular chromosomes, and we are not undergoing crossing over.
Align along the central plate of the cell and subsequently separate, one traveling to each...
Well, review. Okay, so back to metaphase plate here, right? Again, chromosomes lined up. That was a better line. Chromosomes lined up at the equator, and it is random as to whether the maternal is pointing to one pole or the other. And again, this is acting to shuffle the genes, not happening in mitosis, right? In mitosis, there's no homologs, and we're ending up with just producing identical cells.
Of the cell and subsequently separate, one traveling to each end of the cell. In meiosis, a second division sequence occurs, resulting in 4 cells with half the number of chromosomes. Mitosis involves a single division sequence, resulting in 2 cells with no net change in the number of chromosomes.
All right, so I mean that summarizes it in a nutshell, right? The fact that there's 2 divisions in meiosis versus 1. So the major events that we're seeing in mitosis and meiosis, and again, the very important crossing over, this random shuffling of genes, producing offspring that's not identical to the parents, and at synapsis, the tetrads are forming, they're pairing up along their lengths, they're crossing over to swap genes, and that's occurring in prophase 1. So major advantages of sexual reproduction is diversity. We've mentioned that a lot. I've said those words a lot. Important to know, diversity is important. It's more variability. It's providing new combination of traits so that the offspring can survive and adapt to changes in its environment. So meiosis is important for genetic variation. We're not getting the exact same combination as our parents are. And remember the video we just showed that showed 3 different chromosomes. So if we had 2 to the 3 we would have 8 different possible outcomes. Well, we have 23 chromosomes in each cell, right? Because we're talking making sperm and egg. So this random assortment is providing many millions of different combinations in each gamete. And that, in addition with the prophase 1 crossing over, and then the next event that I want to talk about, which is called random fertilization, all of these things together producing a unique offspring. So you have random sperm fertilizing a random egg, and that unique zygote that you are making, well, you're 1 in a million, right? You might look like your parents and share some characteristics. Maybe you look more like dad or more like mom, but you are your own unique individual, and that's the point of reproduction and meiosis. So here's these 3 things. This is the last time I'm going through that increased genetic diversity, crossing over, occurring in prophase 1 between the homologs, and independent assortment happening in meiosis 1, that is producing the different alignment of paternal and maternal chromosomes. And then that third one we just mentioned, not in any stage of meiosis, that's after meiosis has occurred, but random fertilization, where an egg and ovum is going to be fertilized by a random sperm. So that's pretty much it. That's kind of a summary. The point of meiosis is that we are producing variability, diversity. We're producing offspring. Yes, they're somewhat similar, but they're also different to ensure survival of the species. So that's it. That's all. That was meiosis. And again, if you have any questions, michelle.ledeen@ghsd75.ca. Talk to you later.